A warm welcome to everyone in attendance today, and a special thanks to all of our speakers. We know that your commitment to patients and the mission to uncover novel, impactful care solutions on their behalf is a time-intensive pursuit, and we value that you've taken time from your day to join the Charisma team and our partners. While I am unable to join you live today, I've been looking forward to this discussion because it offers a unique opportunity for our team of healthcare providers, researchers, and scientists to dive in deep on our clinical trial findings, discuss the future of our studies, and engage directly with you, care providers and scientists on the front lines of cancer care. Today, you will hear from Charisma leaders and collaborators about a variety of engineered myeloid topics, predominantly our ongoing phase 1 first-in-human clinical trial study of CT-0508 and immunotherapy leveraging CAR-Macrophages in patients with HER2-expressing solid tumors. We will kick things off today with our Chief Scientific Officer, Dr. Michael Kliczynski, who will set the scene for our work to date. As the co-founder of Charisma Therapeutics and the co-inventor of the CAR-Macrophage technology, Michael's experience and leadership has been a constant North Star for our team and motivated our continued scientific investigation of macrophages and other myeloid cells as weapons against hard-to-treat cancers and other serious illnesses. We'll take you through our most recent data accepted for presentation at this year's ASCO, as well as observations of the therapy thus far. While the primary endpoints of this phase 1 study are focused on providing safety and feasibility, we will also preview for you the clinical observations beyond this. Throughout our presentation and discussion today, we hope that you'll learn more about the technology's origin story, what inspires us about myeloid cells, particularly the macrophage, and how we're leveraging our key learnings elsewhere. Before I hand it back to the team, I'll close with a few thoughts that I hope will further ground you in the work we're doing at Charisma. From preclinical data to dosing our first patients, Charisma has been steadfast in our commitment to the understanding and harnessing the potential of engineered macrophages as a viable therapeutic option for patients. While our current trial is focused on hard-to-treat HER2-expressing cancers, our investigative efforts beyond this are well underway. It has always been the vision of this scientific endeavor to not just understand the power of engineered monocytes and macrophages, but to tap into and unleash the potential of these therapies in combination with other programs. We believe deeply in the power of partnership in the field of immunotherapy. In collaboration with best-in-class pharmaceutical and biotech companies like Merck, Moderna, and Novartis, we are mapping out preclinical and clinical trial programs that evaluate what bringing technologies together can achieve in the field of immunotherapy. I'm excited for today's discussion and the work that lies ahead. I thank you again for your time today, and am proud to be part of the Charisma effort to innovate in the name of science and on behalf of real clinical needs.
Good afternoon. Thank you everyone for joining our webinar today. My name is Michael Kliczynski. I'm the co-founder and chief scientific officer of Charisma Therapeutics. And today we'll be giving a presentation around our CAR-Macrophage platform. The speakers you'll hear from today are myself and Ramona Swaby. Ramona is a medical oncologist and is our vice president of clinical development. Our agenda today will cover 3 key areas. The first is around the CAR-Macrophage platform. I'll provide an overview of the CAR-Macrophage technology, the role of myeloid cell therapy in oncology. I'll provide an overview of Charisma's proprietary platform technology. We'll go over preclinical data, including both published and unpublished data. And I'll describe the CAR-Macrophage manufacturing process that is currently being utilized in the manufacturing of CT-0508, which is our lead program, an autologous anti-HER2 CAR-Macrophage that's currently in a phase 1 study. CT-0508 represents the first genetically modified macrophage that has been administered to man, and it's the first CAR-Macrophage in a clinical trial. Ramona will present this section and will describe the phase 1 study design. She'll describe the patient population to date. She'll present the safety and feasibility data that we've seen so far, and we'll do a deep dive into the clinical outcomes, particularly on the translational side, where we've seen exciting data in the tumor microenvironment demonstrating and validating the mechanism of action of the CAR-Macrophage approach in the solid tumor setting. Lastly, I'll describe some of the next generation approaches and the pipeline being developed by Charisma Therapeutics, including a combination study with pembrolizumab, a PD-1 blocking agent, advances in myeloid cell synthetic biology, including novel approaches and novel chimeric antigen receptor designs. I'll describe Charisma's CAR monocyte program, allogeneic iPSC-derived CAR-Macrophage program, and I'll describe a novel approach that we're developing in partnership with Moderna Therapeutics to develop in vivo reprogrammed myeloid cells using LNP and mRNA technology. We will have a Q&A session at the end. You should see a Q&A box in your browser, and you can feel free to type in questions at any point. And at the end of the presentation, we'll go through as many questions as we have time for. Now, as many of you know, CAR T cells have been tremendously effective in leukemia, some lymphomas, and increasingly in multiple myeloma, but responses in solid tumors have been elusive. Regardless of the CAR target, the type of CAR T cell, the mechanism of engineering the CAR T cell, the signaling domain or domains, etc., we have seen limited efficacy in solid tumors. And while we don't know exactly which barriers are key for overcoming this resistance to CAR T in solid tumors, the field generally agrees that there are three categories. The first is trafficking and penetration. Solid tumors do their best to limit lymphocyte infiltration. The second is the tumor microenvironment, and broadly speaking, the various immunosuppressive challenges that the TME imposes upon T-cell-based therapies. And the third, which I think is critical and often overlooked, is target antigen heterogeneity. Solid tumors are highly heterogeneous in their antigen expression, both within a single tumor mass and across multiple metastatic sites. This allows for inherent resistance, even if your cell therapy overcomes the trafficking barrier and overcomes immunosuppression in the TME. If you target a single antigen, you'll rapidly select for antigen negative or antigen dim target cells, and you'll have a tumor that grows out rapidly. Now, our approach at Charisma Therapeutics is to utilize a different cell type, a myeloid cell. And we believe that by using monocytes and macrophages, we can address all of these issues directly. First, myeloid cells are actively recruited to solid tumors. Myeloid cells are the most abundant immune cell infiltrating solid tumors the majority of the time in the majority of solid tumor types. We demonstrated that we can engineer human monocytes and macrophages with chimeric antigen receptors to target macrophage effector function through a chimeric antigen receptor. Now, macrophages act through unique mechanisms. They don't kill through lysis, such as T cells do. They largely kill through phagocytosis, and they can also induce apoptosis of tumor cells through the release of various factors such as TNF-alpha and others. We demonstrated that by putting a CAR on a macrophage, we can guide macrophage phagocytosis, and as long as the target antigen is expressed on the tumor cell, the macrophage will phagocytose and kill the target cell. Charisma's proprietary methods of manufacturing CAR-Macrophages enable a product that is not only targeted, but M1 polarized and restricted to an M1 phenotype, such that when the CAR-Macrophages enter the tumor, they not only induce a pro-inflammatory environment, but they also resist subversion to an M2 phenotype. We found that CAR-Macrophages, upon binding antigen, secrete a variety of cytokines and chemokines that activate surrounding immune cells, produce an inflammatory environment, and directly recruit T cells, dendritic cells, NK cells, and activate bystander macrophages from an M2 to an M1 phenotype. Critically, while the direct killing function of CAR-Macrophages is targeted, these are professional antigen-presenting cells, unlike T cells and NK cells, and these professional antigen-presenting cells will, after CAR-mediated phagocytosis, will consume tumor-derived antigens and prime a T cell response, leading to epitope spreading and a broad adaptive immune response that can be effective against target negative or target dim tumor cells. And this leads to an expansion of the effect, as well as a long-term persistence of the effect through T cell memory. I'll go through some preclinical data that describes several points that I highlighted here in the mechanism of action. Now, as I described, macrophages are abundant in solid tumors. There's ample data across a variety of solid tumor types demonstrating that macrophages are very abundant in the TME. However, that doesn't guarantee that adoptive transfer of these myeloid cells, whether monocytes or macrophages, will lead to tumor infiltration. We have demonstrated in preclinical studies, as well as several other groups, have demonstrated that intravenous administration of macrophages does lead to tumor infiltration. Now, a significant challenge existed in this field, which is that macrophages are difficult to engineer. The commonly used viral and non-viral means that are used to generate, say, CAR T cells or CAR NK cells, notably lentivirus or retrovirus, are simply ineffective in the transduction of human myeloid cells. Charisma identified a unique viral vector that is very efficient in the transduction of human myeloid cells called Ad5F35, and Charisma has also in-licensed the only other known viral vector that is efficient in the engineering of human myeloid cells, which is VPX lentivirus developed by Ned Landau at NYU. Charisma has also developed several additional methods to engineer human myeloid cells with non-viral tools, such as modified mRNA, which is useful if one wants short-term expression of their payload. For the remainder of the talk today, I'll focus on our lead program, CT-0508, which utilizes Ad5F35 as the vector to engineer human macrophages. Now, We identified Ad5F35 as a unique tool to engineer human macrophages ex vivo and found that it has an interesting 2-in-1 effect. In addition to delivering the chimeric antigen receptor to the macrophages, this vector is very efficient in transduction of this cell type. It also activates the macrophages to a so-called M1 or pro-inflammatory phenotype. In the middle panel here, you can see CAR expression after transduction with Ad5F35 showing approximately 88% CAR expression. I'll go through and show the transduction efficiency in our phase 1 study in a later slide. On the right-hand side is an RNA-seq experiment, each column representing a patient, or a donor in this case, and we're looking at 50 different M1 associated genes, and you can see that in the CAR population, all of these M1 pro-inflammatory myeloid genes are activated at the end of our manufacturing process. We have demonstrated furthermore that upon antigen encounter, the CAR-macrophage further upregulates pro-inflammatory M1 associated genes. Now, what do CAR-macrophages actually do? The core hypothesis that led to the development of CAR-macrophages by myself and Sargila Carl June back at the University of Pennsylvania was the concept of redirecting phagocytosis. Can we tell these macrophages what to do by putting in a CAR? This video that's been playing while I've been speaking, it demonstrates CAR-macrophages in red, phagocytosing tumor cells that are in green. This occurs in the absence of any additional opsonization, so we don't need to add antibodies, we don't need to block the CD47 do not eat me signal, etc. Simply adding a CAR and ensuring that the tumor cell expresses the antigen is enough to drive robust macrophage activity. Now, in addition to phagocytosis, when the CAR binds to its target, it also has several other functions. So the first graph on the left is a cytokine release assay where we evaluated either untransduced macrophages, delta zeta CAR macrophages, which lack the CD3 zeta signaling domain that we use in our chimeric antigen receptor, or a full CAR macrophage that was either triggered with a control protein or a HER2 antigen. And you can see that upon antigen encounter, only the full CAR induces release of TNF-alpha, which is a pro-inflammatory cytokine that has direct anti-tumor activity as well. A variety of other cytokines are released that aren't shown here, chemokines as well. In the middle panel, we demonstrate quantitatively that phagocytosis is targeted. Macrophages with a CAR against HER2 do not phagocytose HER2 negative tumor cells in the middle, but they do phagocytose HER2 overexpressing cells such as SCOF3s on the right. Together, these effects of phagocytosis and TNF-alpha release lead to a potent direct killing ability of the CAR macrophage. On the right-hand side is a killing assay where we co-culture untransduced control macrophages in green or CAR macrophages in red with HER2 overexpressing tumor cells that express GFP. And we track this live with the IncuSite video microscopy tool. And you can see that CAR macrophages directly clear all the tumor cells at even low effector to target ratios such as 1:1. We previously evaluated human CAR macrophages in immunodeficient xenograft models. That data was published in Nature Biotechnology in 2020, and we didn't include it here, but I'll summarize it by saying that we demonstrated that human CAR macrophages control tumor growth and improve overall survival in multiple xenograft models that are immunodeficient, demonstrating that CAR macrophages themselves, through their direct killing function, can improve outcomes. However, we recently established fully immunocompetent syngeneic models to allow us to evaluate changes in the tumor microenvironment, as well as to evaluate critically impact on the adaptive immune system. Toward that goal, we established a HER2 overexpressing CT-26 model, which grows in a wild-type BALB/c mouse. We demonstrate that CAR macrophage monotherapy in red improves tumor control and significantly improves overall survival of the Kaplan-Meier curve on the top right. And critically, as these mice are fully immunocompetent, we can evaluate changes in the T cell compartment. On the bottom left is representative immunohistochemistry, looking for murine CD8 T cells infiltrating the CT-26 tumors. You can see control tumors or control untransduced macrophage treated tumors have very few T cells, but once CAR macrophages are administered, there's significant influx of CD8 T cells into the TME. And we further quantified this with flow cytometry and found that CD8 T cells, CD4 T cells, not shown here, NK cells and dendritic cells are all increased in the tumor after therapy. And critically, we don't simply want to recruit T cells for the sake of having more T cells. We want to activate tumor-reactive T cells. And we quantified tumor-reactive T cells by looking for activated interferon gamma positive, CD8 positive TILs. and found that there is a striking increase in the fraction of activated TILs in the tumor after CAR-Macrophage therapy. To put this to the test, to directly evaluate this concept of epitope spreading, we performed 2 studies. First, we performed a dual flank study designed to look for what's known as an abscopal effect. Here we grew a HER2 positive and a HER2 negative version of the same CT-26 tumor. These tumors are genetically identical other than for the HER2 antigen. On the left-hand side, on the bottom left, you can see we treated the HER2 positive tumor with an intratumoral injection of CAR-macrophages or untransduced macrophages. The CAR-macrophage treated tumors rapidly melted, but notably, the wild-type HER2 negative tumors on the contralateral flank demonstrated a significant reduction in tumor growth rate, suggesting that an abscopal effect is occurring and T-cell immunity against secondary antigens or neoantigens is being induced. We evaluated the tumor microenvironment of that HER2 negative tumor and similarly found an increase in the infiltration of a variety of immune cells, including T cells, dendritic cells, and NK cells. Now, to take this a step further and to evaluate a more realistic issue, which is antigen-negative relapse, which we already know is a meaningful challenge with CAR T cells in blood cancers, where antigen heterogeneity is much less of an issue than in solid tumors. We performed the following study. We grew HER2 positive tumors and treated them with anti-HER2 CAR-Macrophages. We then took the complete responders that cleared their tumor burden and re-challenged them with a HER2 negative version of the CT-26 tumor. At the same time, we challenged naive mice with no history of therapy with wild-type CT-26, and we tracked tumor growth and survival. We found that even 2 months after clearing their tumors, these mice that were treated with CAR-Macrophages were protected from re-challenge with a tumor that lacks the dominant antigen that the CAR is targeted to bind. And all of the naive mice died from disease in approximately a month, while all of the previously CAR-Macrophage treated mice were protected, tumor-free, and alive. Not shown here, but we demonstrated that this is in fact mediated by memory T cells that are instilled by CAR-Macrophage therapy. So with that data in hand, Carisma entered the clinic with our lead program, CT-0508. This is a HER2-targeted CAR-Macrophage. It's an autologous cell product. The phase 1 study is ongoing. The early data that Ramon and I will go through supports safety and validates the mechanism of action clinically. We currently have 5 clinical sites that are open for screening and enrollment, which are listed here, and several other sites will be open in the near future. The FDA granted Carisma Fast Track designation for the CT-0508 program in September of 2021. CT-0508 is comprised of what is considered a first-generation CAR in the T-cell world. It comprises a humanized anti-HER2 scFv that's linked through a CD8 hinge and CD8 transmembrane domain to CD3 zeta. CD3 zeta is not normally found in macrophages. It is part of the T cell receptor complex and is utilized as the primary signaling domain in essentially all CAR T cells. But having screened hundreds of signaling domains at Carisma, we have found perhaps the surprising result that CD3 zeta is the single most potent phagocytic signal for a macrophage, likely driven by the fact that it is the most ITAM domain-rich immunoreceptor. Now, in order to enter into the clinic, Carisma developed a full-scale GMP process that I'll walk you through. The source of the cells are autologous mobilized peripheral blood monocytes. The process starts with monocytes. Patients first sit for get mobilization with G-CSF or Neupogen, which is an at-home subcutaneous injection that increases the peripheral blood count of monocytes and enables an increase in the potential dose. G-CSF is well tolerated and has been approved for quite some time. Patients then sit for a standard apheresis. A leukopak is collected and shipped fresh to our manufacturing site, which currently is Miltenyi, but Carisma has recently entered into a partnership with Novartis collaborating with Carisma to develop to manufacture CT-0508. We use the Miltenyi Prodigy instrument, which allows us to select CD14 positive monocytes with positive selection. This is a rapid and automated process. Monocytes are selected and are seeded for differentiation into macrophages, and we utilize GM-CSF, which generates a pro-inflammatory-like macrophage that has antigen presentation qualities. The macrophages are transduced with the Ad5F35 vector, which delivers the chimeric antigen receptor, and at the same time polarizes the cells toward an activated or M1 phenotype. The entire process takes 1 week and it is mostly automated, At the end, the cells are cryopreserved, and they're shipped back to the clinic in a cryopreserved state, where similar to CAR-T, they're thawed at the bedside by the nurse and administered intravenously over approximately 30 minutes. Notably, CAR-T and CAR-NK currently all require chemotherapy conditioning, typically with Cy/Flu. CAR-Macrophage therapy does not require any chemotherapy conditioning prior to administration, and our ongoing study does not incorporate any chemotherapy. It is true monotherapy. Now, in the years of developing the process, we utilized healthy human donors to develop the process and go through process development, engineering rounds, and so on. And we knew going into the study that cancer patients are different. There may be quantitative differences to their myeloid compartment. There may be qualitative changes to their myeloid compartment. We're happy to report that so far we've had no challenges in manufacturing autologous CT-0508 from very heavily pretreated metastatic solid tumor patients. Our key release criteria on the left include viability. We routinely see high viability in our cell product. Purity, defined as the percentage of cells that express a macrophage marker. The purity is underestimated in this graph, as we found that this particular marker that we use to define macrophages is downregulated as the macrophages become more M1 polarized. The CAR expression is routinely high, ranging from about 75 to about 95% CAR positive. We evaluate each batch of CT-0508 for information only in several functional assays in vitro. And in the middle panel here is an in vitro tumor cell killing assay demonstrating that CT-0508 are able to, patient-derived CT-0508 are able to kill HER2 overexpressing breast cancer cells in an IncuCyte-based killing assay. We also characterize each product with flow cytometry, single-cell RNA sequencing, and a number of other deep characterization assays. And briefly shown here is that the average CT-0508 product upregulates an unbiased M1 gene panel as compared to the baseline product. So the cells are in fact M1 polarized. With that, I'll pass over to Ramona Swaby, who again is a medical oncologist and our Vice President of Clinical Development, and Ramona will walk you through the clinical section.
Thanks, Mike. So this is the study design for the first-in-human, first-in-class CAR-Macrophage clinical trial. And the study seeks to enroll 18 patients total in 2 cohorts. The first cohort of 9 patients received fractionated dosing of CT-0508 given on separate days. And the second cohort of 9 patients received CT-0508 as a single infusion given on day 1. Patients are treated with 4 days of GCSF, as you heard, to increase bone marrow monocyte production prior to apheresis, again without the need for lymphodepletion, with a cytotoxic-based preparative regimen. Now, patients undergo serial biopsies and blood work collection for translational studies, pre-treatment at baseline, as well as on day 4 and day 8 and week 4. And then the first RECIST tumor assessment occurs at week 8. The primary study objectives are safety and tolerability of CT-05 administration, as well as manufacturing feasibility of a pure product. And the secondary objectives include cellular pharmacokinetics, preliminary assessment of efficacy, and immune correlates, including serum cytokines, evaluation of the tumor microenvironment, and T-cell repertoire analysis. I'm pleased to share with you that the cohort 1 has just completed accrual, and we are actively enrolling cohort number 2. And with the data that I'm going to share with you in the next couple of slides, I hope to be able to demonstrate that to date, CT-0508 has been able to be successfully manufactured and safely administered to patients and is associated with a favorable safety and tolerability profile. Next slide, please. So this is the data from the first 7 patients that have been enrolled. And the patients enrolled, you can see, are consistent with a population typical for a heavily pretreated phase 1 oncology population, with a median age of mid-60s, an even distribution of male and female patients, a preserved ECOG performance status as required per the eligibility criteria of the protocol, and all patients to date have been confirmed to be HER2 overexpressing as required per protocol. As you can see, the patients that have enrolled have a variety of tumor types that were eligible for enrollment based on HER2 overexpression, including tumors with GI tumors, ovarian cancer, and salivary duct carcinoma, in addition to breast cancer. The patients are heavily pretreated with a median number of prior therapies of 2, median number of prior anti-HER2 therapies, of 2, and a median number of prior cancer therapies of 3. But you can see the range is quite extensive, extending to as high as 11 prior cytotoxic regimens and as high as 9 prior HER2 directed therapies. I just like to circle back. You can see with the number to the broad sort of variety of tumor types that enrolled, and this potentially speaks to the broader clinical applicability of the CAR-Macrophage program, as well as our upcoming HER2 CAR monocyte program as well. Next slide, please. The median number of cells were over 1 billion that have been infused, with a range approaching up to 5 billion. And I think this demonstrates that CT-0508 was able to be feasibly administered in a time frame and volume that make it attractive for both patients and providers in terms of infusion chair time, and hopefully also make it feasible to be able to be administered in combination with other therapies. Next slide, please. The safety profile in the first 7 patients treated thus far has been very encouraging. There have been no DLTs. 5 of 7 patients have had low-grade CRS, cytokine release syndrome, either grades 1 or 2. 4 of 5 patients had grade 1 CRS, and 1 patient had grade 2 CRS, characterized by fever and hypotension, which resolved within 24 hours with Tylenol and IV fluids. We've had no ICANS reported to date, and with respect to serious adverse events or SAEs, there has been 1 subject who experienced 2 SAEs of CRS and an infusion reaction, which all resolved within 24 hours. 2 additional subjects have had SAEs that have reflected progression of their disease. So the first patient is an esophageal cancer patient who had hemorrhaging of the enlarging primary lesion at the time of his progression of disease. And the second patient had lymphangitic spread of their breast cancer at baseline when they entered the trial, and experienced dyspnea associated with progression of their lymphangitic pulmonary metastases. There have been no major organ toxicities observed. And with the next slide, you'll be able to appreciate that the overwhelming majority of the adverse events are grades 1 and 2. So as you can see on this slide, there are very few grade 3 adverse events and only 1 grade 4 adverse event. CRS, again, grades 1 and 2. And on the left side of the slide, you can see that the most common adverse events were laboratory abnormalities that included decreased lymphocytes, white blood cells, and anemia, as well as some decreases in electrolytes and albumin. In the next slide, here you can see that the decreases in the bone marrow cells and platelets are transient and self-limited and resolve within approximately 48 hours. Next slide, please. In this slide, blood work for cytokine analysis was collected, as Mike had said previously, as part of a sort of broad complement of translational assays assessed from subjects on study. And what you can see is that there is a transient elevation of body temperature that correlates with minor and transient increases in serum IL-6 levels that self-resolved within 48 hours. And finally, In the next slide, we have early efficacy results that demonstrate that we have stable disease in 4 of 7 patients who had a tumor assessment at 8 weeks. With that, I'll stop there and pass it back to Mike.
Okay, thank you, Ramona. So now we'll go into some of the translational data from the peripheral blood and the tumor biopsies of these patients. First, looking at the peripheral blood kinetics of CT-0508, we found that in approximately 30 minutes after intravenous infusion, as expected, the cell number peaks. And within approximately 6 to 8 hours, CAR-macrophages are no longer detectable in the peripheral blood, not representing their persistence. We know that from preclinical data that they can persist for months, but demonstrating that these cells don't continue to recirculate in the peripheral blood. They traffic into tissues. And on the right-hand side, we evaluated infiltration into the TME using RNAscope with a unique probe against the mRNA, encoding a unique sequence of the chimeric antigen receptor that's not found in the human genome, so we can identify them with specificity. And we were able to detect CT-0508 in the TME of 6 out of 6 patients that have completed analysis of this assay. In some patients, we were able to detect, or I should say in 5 out of 6 patients, we were able to detect the cells at day 8 post-infusion. And in some patients, as you can see in this table, we were able to also detect them in week 4. In all patients in totality, we were able to detect the presence of the cells at least one time point, and of course, not prior to therapy. We evaluated the tumor microenvironment using single-cell RNA sequencing. We were fortunate that in a number of our patients, we were able to get paired biopsies at 3 time points at screening prior to therapy, at day 8 after the last dose of CT-0508 to look for early changes in the tumor microenvironment, and at week 4 post the last dose to look at changes that take time, such as induction of adaptive immunity. Using single-cell RNA sequencing allows us to evaluate the fraction of a variety of immune cells and also their gene expression to look for patterns in certain programs. First, we found that there was an increased fraction of M1 macrophages in the tumor microenvironments of 3 out of 3 patients where the paired biopsy single-cell RNA-seq data was available. You can see in subject 1, there was a significant increase from about 40% to 60%. In subject 2, there was a mild increase, and in subject 4, there was a notable increase from approximately 7% to approximately 30% of macrophages being M1 based on an unbiased M1, M2 score from the literature. Zooming in on the myeloid compartment of the first patient, this was an esophageal cancer patient. Here we're looking at the gene expression of the intratumoral myeloid population. This bubble plot can be interpreted by looking first at the size of the bubble. The larger the bubble, the greater the percentage of the myeloid cells express that gene. And the warmer the color, the greater the intensity is of the gene expression. Here we're looking at a number of pro-inflammatory so-called M1 genes, such as TNF-alpha, IL-6, IL-1 beta, IL-1 alpha, and so on. These genes are being induced by CT-0508 therapy in the myeloid compartment within the tumor microenvironment. Now, moving to the T cells, as I described in the introduction, this is an antigen-presenting cell therapy. And the ultimate goal is to drive robust T cell immunity. And we found that CT-0508 induced significant T cell infiltration into the tumor microenvironment based on single-cell RNA-seq data. First, if you ignore the different colors that comprise each bar, you can see that overall there's a significant increase in the frequency of effector T cells in the tumor microenvironments. Then if you look at each color individually, breaking down the T cells by T cell subtypes, you can see that there are differences across these 3 patients in the subtypes of T cells that are increasing in the tumor. But the overall pattern is there that CD8 effector memory T cells are increasing, activated CD4 T cells are increasing, proliferating T cells overall are increasing. And in one patient, subject 2 here, you can see a significant increase in the fraction of exhausted CD8 T cells as well. Now, to dive deeper into the T cell data, we utilize the adaptive platform, which sequences T cell receptors and allows to look for changes in the T cell repertoire, changes such as T cell clonality and so on. And you can track unique T cells over time as well. So we sampled blood prior to therapy and at day 15 post-therapy. And the plot on the left-hand side is known as a Morosita plot. And what we're looking for here are changes compared to baseline. Any data point that you see in orange represents a T cell clone that was previously either undetected or a T cell clone that has significantly expanded. Any clone that is found on the left y-axis represents a brand new T cell clone that was previously undetectable prior to therapy. If you look across all subjects shown here, there is a significant increase in both total expanded T cell clones and newly detected T cell clones as compared to screening in every patient that has been treated. Now, to actually evaluate whether these T cells are tumor reactive, we evaluated several further components here. So first, focusing on the treatment emergent new T cell clones, which represent T cell receptors that were previously undetectable prior to therapy. We mapped these T cell receptor clones against the TCR sequencing performed on the tumor biopsies. And we found that in each of these patients where this 4 patients where the study was performed and completed analysis, there was an accumulation of newly emergent T cell clones in the tumor microenvironment over time, suggesting that there is tumor reactivity of these clones. You'll see notably, subject 4 had a striking increase in their in the accumulation of these newly expanded T cells in their tumor by week 4. Now, to attempt to evaluate the actual phenotype of these tumor-infiltrating newly detected T cell clones, we mapped the TCR sequence of the most prevalent T cell clone in the week 4 tumor microenvironment of the first patient with esophageal cancer, and mapped it against the single-cell RNA sequencing data to evaluate the identity and gene expression of this most prevalent clone. And we found that compared to all other clones, this particular T cell clone was highly activated and took on a cytotoxic CD8 T cell phenotype, expressing genes that are known to be involved in tumor cell lysis, such as PRF1, which is perforin, GZMB, which is granzyme B, and other genes associated with a cytotoxic activated CD8 effector cell phenotype. So taken together, these data suggest that CT-0508 are priming an anti-tumor adaptive immune response. So I'll stop there with the translational data. In summary, Ramona and I have shared data that demonstrates the feasibility of CT-0508. CT-05 was successfully manufactured from autologous mobilized monocytes. The patient product demonstrated high CAR expression, purity, viability, M1 polarization, and confirmed functionality in vitro. The preliminary clinical profile demonstrates a very clean safety profile. There have been no dose-limiting toxicities to date. There have been no adverse events that led to dose modification or discontinuation. We have not seen severe cytokine release syndrome. We have seen mild CRS, grade 1 or grade 2, that was self-limiting. We have not seen any neurotoxicity, and we have not seen any major organ system toxicity and no sign of toxicity that is associated with on-target off-tumor toxicity against the target antigen HER2. Any increases in serum cytokine levels were transient and were also self-limiting. Lastly, we've seen confirmation of the mechanism of action. We've seen tumor infiltration of CT-0508. We've seen increased infiltration of CD8 T cells and M1 macrophages after treatment. We've seen, broadly speaking, activation of the tumor microenvironment. And based on tracking T cell receptor clonality, we've seen data suggesting that there is induction of an adaptive anti-tumor immune response. So to summarize how we see the key characteristics of CAR-M in oncology, first, CAR-Macrophages do not require chemotherapy conditioning. CAR-Macrophages, at least the CT-0508, does not cause severe CRS or neurotoxicity. Macrophages do not cause graft versus host disease, as they don't have T cell receptors, and macrophages can thus be used as an allogeneic off-the-shelf product. There's potential for allogeneic compatibility. And importantly, macrophages do not cause immunosuppression. From a mechanism of action point of view, CAR-Macrophages infiltrate solid tumors. By using viral vectors with durable transduction, we see durable CAR expression. We see direct cytotoxicity through phagocytosis and other means. We see that CAR-Macrophages are able to remodel the TME, recruit T cells, and present antigen, lead to epitope spreading. And as I'll show some data shortly, CAR-Macrophages can sensitize checkpoint inhibitor-resistant tumors to immunotherapy. From a manufacturing and commercial potential point of view, CAR-Macrophages demonstrate feasible manufacturing. The process is largely automated. The cells can be cryopreserved and in fact are stable in a cryopreserved state for very long periods of time. The process is fairly rapid for an autologous program. Our manufacturing cycle takes 7 days, with the majority of the time being hands-off, and this process allows for a reduced cost of goods. I'll talk about some of our next approaches, which further improve upon these factors. Now, Carisma is working on a variety of novel approaches, really with two goals in mind. The first is to enhance activity and innovate the field of myeloid cell therapy, and the first is to improve the manufacturing. I'll touch on a few components today. At Carisma, we're working on combination therapies. We've enabled gene editing of human myeloid cells with CRISPR and have exciting data with SIRP-alpha knockout CAR-Macrophages. We have developed novel proprietary chimeric antigen receptor designs that further enhance and augment macrophage function. Carisma has also developed a CAR-Monocyte program, which has the capability to shorten the manufacturing cycle from a week to a day. And using our vectors, we have a durable CAR expression and durable persistence of the cell product. Carisma has partnered with Dr. Bruce Blazer, a pioneer of iPSCs out of the University of Minnesota, to develop allogeneic iPSC-derived monocyte and macrophage cell therapies. And I'll show briefly some data there. And we have a number of academic partners with whom we're working on these novel technologies and others. So first, to talk about combination therapies. Given the mechanism of action that I described, which is TME activation, T-cell recruitment, and T-cell priming, it is certainly logical to anticipate that having a non-exhausted T-cell population is likely to enhance outcomes with T-cell priming. We've seen first in preclinical data on the left-hand side that in tumors that relapse after CAR-Macrophage therapy, the PD-1, PD-L1 pathway based on RNA sequencing is significantly upregulated. We've seen in our early clinical data that there is an increase in some patients of exhausted CD8 T cells, hand in hand with an increase in CD8 T cell activation. And inhibitory receptors are upregulated on CD8 T cells in patient tumor microenvironments post CT-0508 therapy, likely as a response to the inflammatory effects that CT-0508 has. We also know that CT-0508 themselves are M1 polarized macrophages, and M1 macrophages express PD-L1 as it's also an activation marker on myeloid cells. So we thus began evaluating the combination of PD-1 antagonism with CAR-Macrophages and found in this case in a PD-1 resistant solid tumor model, CAR-Macrophages have monotherapy efficacy, but notably they sensitize the tumors to PD-1, and the combination of the two agents leads to significantly improved outcomes. We are working with Merck to evaluate the combination of CT-0508 with Keytruda or pembrolizumab in a clinical study that should be started in the near future. And we found preclinically that in addition to improved outcomes in terms of tumor control, combination of CAR-Macrophage therapy with T-cell checkpoint blockade also leads to synergistic activation or modulation of the tumor microenvironment. So this is multiplexed immunofluorescence of CT26 HER2 tumors. You can see that control tumors are mostly comprised of blue, which are tumor cells, and green, which are macrophages. PD-1 alone has minimal impact in this model. CAR-Macrophage monotherapy leads to an increased infiltration of macrophages and T cells, which are shown in yellow. But once you give the two agents together, you can see the tumor really lights up, and we see infiltration of dendritic cells at a much higher level. We see a variety of granulocytes, NK cells, and so on coming into the tumor. Carisma has also developed a number of proprietary next-gen synthetic biology strategies, one of which is an area we've been working on for quite some time, which are next-gen myeloid CARs. As many of you, I'm sure, are familiar with the CAR-T space, you know that adding co-stimulation to CAR-T cells was critical for efficacy with either 4-1BB or CD28. And the reason for that is T cells require a signal 1 and a signal 2 to be properly activated. Macrophages do not require two signals. Macrophages are in some ways simpler than T cells, in other ways not. But macrophages can be controlled often with a single signal, a yes-no decision. Do I phagocytose or not? Do I release cytokine or not? And we found that the most potent single signal is CD3 zeta. But we have been evaluating additive signaling. Can additional signaling domains further enhance either the direct killing activity or cytokine release, or perhaps other functions of the macrophage. We have identified dozens of constructs that Carisma is evaluating, and we've seen as an example on the right-hand side that in some cases with some of Carisma's proprietary second-generation myeloid CARs, we can significantly improve the potency of the product. So you can see this is a killing assay at a so-called normal dose. And at a normal dose, you can see that all CARs, regardless of their design, kill all the tumor cells. But if you proceed to what's called a stress test dose, reducing the dose to have mild activity, you can see that one of the second-generation CARs, second-gen CAR-C, significantly enhances the killing activity even at these low stress test doses. The concept that we're pursuing here is introducing signaling domains derived from receptors that are known to engage macrophage activation. An example being the CD40 pathway, which we found is very active in macrophages, as well as MyD88 and other innate immune receptors. Another approach that Carisma has taken is to utilize CRISPR-Cas9 to delete potentially inhibitory checkpoints that are expressed on macrophages to further unleash their function. A perfect example here is the CD47 SIRP-alpha pathway. CD47 is known as the do not eat me signal. Tumor cells overexpress CD47, and there are a number of CD47 blocking or SIRP-alpha blocking agents in the clinic that have demonstrated activity. The approach that Carisma has taken is to utilize CRISPR to delete SIRP-alpha from the CAR-Macrophage to make these CAR-Macrophages essentially blind to the do not eat me signal. And what we found is that we found that we can engineer human CAR-Macrophages and use CRISPR to delete genes with high efficiency with a protocol that was developed by Carisma's scientists. As you can see here, SIRP-alpha is knocked out with very high efficiency, over 95% knockout, and these edited cells are healthy and viable. On the bottom is a phagocytosis assay. The higher the line goes here, the more phagocytosis that occurs. First, untransduced macrophages don't demonstrate any phagocytosis. By simply knocking out SIRP-alpha without introducing a CAR, phagocytosis still doesn't occur. Simply taking your foot off the brakes isn't enough to drive eating. You have to simultaneously bind to the target cell and essentially press on the gas for phagocytosis to occur. CAR-Macrophages phagocytose CD47 high tumor cells, but if you knock out SIRP-alpha, you can see the level of eating significantly and synergistically increases. Now, on the manufacturing front, CT-0508 is a CAR-Macrophage program. Carisma is, in addition to developing CAR-Macrophages, also developing CAR-monocytes. The advantage of CAR-monocytes is largely around the manufacturing process. And Carisma has developed a process in which we first mobilize patients similarly to CT-0508 with GCSF. Patients sit for apheresis, monocytes are purified and rapidly transduced with the Ad5F35 vector, and the cells are cryopreserved. The entire process is automated. The process takes less than 1 day, and it results in a cell product with high viability, high CAR expression, over 90%, and it significantly reduces the cost of goods and the turnaround time, as the manufacturing cycle is reduced from about a week to approximately a day. Notably, by continuing to use Carisma's proprietary viral vectors for this purpose, we see long-term CAR expression and long-term persistence of the engineered cell product, which non-viral means do not result in. As I described, we're also developing allogeneic, off-the-shelf iPSC-derived CAR-Macrophages through a partnership with Dr. Bruce Blazer out of the University of Minnesota. The concept here is to use iPSCs to integrate the chimeric antigen receptor, perform additional necessary edits, create a master iPSC cell bank, amplify the cells, differentiate the cells into myeloid cells through a process that we've developed together with our collaborator, and essentially have a bank of universal iPSC-derived CAR-Macrophages with batch qualification and uniformity that can allow for off-the-shelf CAR-Macrophage availability. This process also is being developed for CAR-monocytes, and we've seen on the right-hand side that we can generate iPSC-derived CAR-Macrophages with significant expansion, roughly 1,000-fold expansion in as short as 19 or 20 days. You can see an example of the CAR-macrophages on the right-hand side derived from iPSCs. They're very uniform, and they are bona fide macrophages based on this GM sustain and extensive characterization of flow cytometry. And we evaluated these iPSC-derived CAR-macrophages in functional assays in vitro and found that similarly to monocyte-derived CAR-macrophages, these cells are functional and they can kill. As you can see, iPSC-derived non-CAR macrophages in yellow have no anti-tumor activity, while iPSC CAR macrophages against HER2 are clearing tumor burden. Lastly, I'd like to touch on an exciting partnership that Carisma entered into at the start of the year with Moderna Therapeutics to develop in vivo LNP mRNA-based myeloid cell therapies. The concept here is as follows. Rather than taking blood and engineering cells derived from patients or taking stem cells, what if we could directly engineer the myeloid cells that are already in the patient and already in the patient's tumor and deliver chimeric antigen receptors to these cells to convert pre-existing TAMs and myeloid cells into tumor-killing cells? Toward this goal, we partnered with Moderna, the world's leader in LNP mRNA technology, to develop these technologies and to generate in vivo CAR-M. The partnership that we entered into with Moderna is a broad R&D partnership for 12 programs in oncology. I'll briefly show one example of proof of concept data in vitro. These are three-dimensional tumor spheroids. Then you can see the video should be replaying. On the left-hand side is a tumor spheroid comprised of breast cancer cells and TAMs treated with a control LNP. On the right-hand side is a spheroid comprising HER2-positive breast cancer cells and TAMs treated with a CAR. And you can see that the TAMs, once they are reprogrammed with LNP mRNA, rapidly lead to melting of this tumor spheroid. The concept, of course, here is to go directly in vivo. These myeloid-tropic LNPs have demonstrated safety and efficient transfection in vivo. Both circulating and tumor-infiltrating myeloid cells are transfected. And we've seen high CAR expression and demonstrated that we can convert macrophages to CAR macrophages using these in vivo, using these LNP mRNA technologies. The beauty of this approach is that it's off the shelf, yet still autologous, meaning you are still engineering a patient's own myeloid cells that have perfect MHC matching to their tumors and can have maximal activity through antigen presentation. With that, Carisma has a pipeline that is primarily focused on oncology, though we have several programs that are in the discovery stage in non-oncology as well. Our ex vivo autologous cell therapy programs comprise CT-0508, a HER2-targeted CAR-macrophage that's currently in the clinic. That's the program we discussed today. A pembrolizumab combination study is coming soon, as well as a regional administration study. A HER2 CAR monocyte study will be opened in the near future. We are also developing CAR macrophages against mesothelin and PSMA. As described, we entered into this broad partnership with Moderna to develop in vivo CAR-M therapies against up to 12 targets comprising both blood and solid tumor cancers. Lastly, we have two programs in non-oncology. The first is around liver fibrosis. I won't go into the details today for the sake of time, but the concept here is to tap into the M2 biology of macrophages using Carisma's myeloid cell engineering toolbox to engineer anti-inflammatory, anti-fibrotic macrophages that infiltrate the liver and simultaneously clear existing fibrosis and reduce inflammation. Lastly, we have a program partnered with, that's being developed together with Sar Gill's lab at the University of Pennsylvania to engineer microglial cells with chimeric antigen receptors to target them to phagocytose protein aggregates for neurodegenerative disease. I'd like to say a big thank you to the team, some of which are shown here, and I'd like to specifically point out Thomas Condamine, who's our head of translational, who was responsible for the beautiful translational data that you saw today from the phase 1 study. And a big thank you to all of the folks on the team who are working hard to enable a first-in-class therapy. So with that, I thank you for your attention.